All right, you guys, so we are going to finish up our genetics lecture with the patterns of inheritance number 2. So this is really going to be what happens if we don't follow Mendelian laws of genetics. So it's a little more complicated than last week, okay? So first we're going to review a little bit about Mendels laws just to remind us about what we talked about last week. And then we're going to talk about the variation of Mendels laws. So what happens if we change it up a little bit, because that's what we actually see in nature. So we do see Mendels laws, but we also see some variation on it that are a little more complex. And then we'll talk a little bit about chromosomal basis of inheritance, linked genes, and sex-linked traits, okay? Meaning, we have our sex chromosomes, and so specifically there are genes on the X chromosome that are not found on the Y chromosome, which means that usually males are more affected than females. So let's review just a little bit. I've included some pictures from the previous lecture on the bottom. So the law of dominance, and we'll talk a little bit more about that because that'll really come into play in some of our variations on Mendel's laws. But if you have a dominant allele, right, if it's in the genotype, then that will be the observable characteristic or the phenotype. So always the dominant allele will shadow the recessive allele when we're talking about true dominance and the law of dominance. So this is different in some of our variation on Mendel's laws. So keep that in mind, and we'll review that a little bit more. And then the law of segregation just means that there's a 50-50 chance you're going to get one allele or the other, right? So say you have on our right-hand side here, we have the gene with the letter H. So you have a dominant H allele and a recessive little h allele. So you have a 50-50 chance of getting either that dominant H, big H, or the recessive little h. So just 50-50 chance, right? It's like flipping a coin. And then the law of independent assortment just means that there's an equal probability of arranging the chromosomes during meiosis, right? So that means that we could get 4 completely separate possibilities when we complete meiosis. Okay, so it just means they can be either on the left or the right side. So we've seen the law of independent assortment a couple of times through meiosis and genetics. And then we'll talk about what happens if we don't follow the law of independent assortment, right? So that means that the genes are going to be linked, so they're closer together on the chromosome, so they actually can't separate, okay? So let's talk a little bit about Mendel's dominant and recessive alleles, just to review, right? So we are talking about 2 alleles existing for every gene, and that may or may not be the case when we talk about some variations. There could be more than just 2 alleles for each gene, but 2 alleles are always going to be assigned to every person of that gene, okay? And alleles maintain their integrity in each generation, meaning there's no blending, meaning that there's no semi-dominant alleles. But we'll actually see what happens if we have some alleles that are both dominant or some blending with heterozygotes. So we'll talk about that. So that could be a variation. And when we talk about those heterozygotes, it's always the dominant allele that is expressed and the recessive allele that's hidden, meaning that the phenotype is always going to express that dominant allele. And so that may not be the case as well when we get into these variations. So what if this were not always the case? right? So on the right-hand side, we have some examples that we'll go through, fruit flies on the top being one when we talk about sex-linked genes. And then this snapdragon plant, which is kind of a pink color, and the parents are red and white. So there's actually a blending of the alleles and of the phenotype. Okay, so let's talk about these. So first, let's talk about incomplete dominance. So when we said that the dominant allele is always going to be expressed in the heterozygote, that's not always the case. Sometimes it does not completely hide that recessive allele in the phenotype. So this is our snapdragon example, where in our heterozygote, the phenotype is somewhere in between the parents. So if we have a red parent and a white parent, then we actually get a blend of the heterozygote. So this just shows you the parent generation, where we have, they're saying R for red, and W for white. So it's a little different than what we've seen before. They're just adding another letter in here for color with the C. And so then you get a CRCW, which is going to be our heterozygote. And that's going to be pink. So the entire F1 generation is actually going to be pink, which is interesting, right? So then our F2 generation, where we actually cross our two heterozygotes, our two pink snapdragons, then we get the parent phenotype to be expressed again, because then we have our red, which will be red, and the white, which will be white. But we still have those two pink heterozygotes, okay? So just kind of something interesting to think about, where there isn't a complete dominance of that dominant allele. The recessive allele does show through in the phenotype. So now what happens if we actually have what's called codominance, where it's kind of an incomplete dominance, so it's a variation of incomplete dominance, where you actually get simultaneous expression of both alleles, okay? So we see this when we talk about blood types in humans. So we have many different blood types, right? We have A blood type, we have B blood type, and then we can combine those with an AB blood type. And then we also have an O blood type. So these letters that we use, A, B, and O, actually is talking about molecules on our red blood cells, right? So our red blood cells are what are circulating in our bloodstream, carrying oxygen, and essentially they're going to have these little molecules sticking on the cell itself. So you can have either A-type molecules or B-type molecules sticking on the cell, or you can have a combination of both A and B molecules. Or you have O, which means you have no molecules on your cells. So those red blood cells are naked, essentially, okay? So what do we mean by codominance? Well, remember that AB type, right? So that means both A and B are codominant. They're both dominant, okay? So that's why in our individuals that have AB blood type, they're both expressing the A and the B type molecules. So again, here are our examples, because we can have an A allele and a B allele, right? So they're codominant. So you can either get your AA individuals or BB individuals, which we just call A type and B type. And there are homozygotes, right? So the alleles are matching. And then we have those AB people that are heterozygotes, but they express both A and B alleles. Okay, and then you get your O, which has no molecules, and then you might think, okay, well, how do we get that, right? Well, we have, so that means we have neither A or B alleles. Well, that's kind of interesting, right? So this is how we answer the question of how do we get the O-type blood. So it's called multiple alleles, and many alleles can exist for one gene in a population. So even though an individual person always is only going to have 2 alleles for a given gene, there are many more that could exist. So that gives you a lot more options, right? So how do we show this multiple alleles when we're talking about the blood types in humans? So we add another letter. So capital I or big I is going to be another allele, or essentially I is going to be an allele. So the capital I with superscript A, superscript B, those guys are the dominant alleles. Okay, so you either have IA or IB, and those are both dominant. Okay, or you can have a little I, which is a recessive allele. And so if you have little I, that's how you get O. Okay, so here's just showing all the possible allele combinations in humans. Okay, so if we have our 3 different allele options where we have big I A, big IB, and little I, we can combo those with another allele, with one of the other 3, right? So if we work our way through this little table, it's just showing you all the individual options that a person could have, because we're always going to only have 2 alleles, right? So if we work our way through, if a person has a blood type A, it's always going to have the big I with the big A, and then another big I with a big A, so that's the dominant homozygote, right? Or you could have a big I, big A with a little I, because that's going to be dominant over the little I. So that is your dominant A overshadowing the little I. So you're still going to be phenotype A, but you're going to have a carrier of that recessive allele. Okay, so those are your 2 options for A type. And that's very similar to B type, right? So to be B, you either have 2 alleles of the dominant IB, or you have a dominant allele IB and a recessive I. Okay, so those are your 2 options there. And then for AB, that means you have one of each of those dominant alleles, right? So you have an IA and an IB. So therefore you are AB type blood type. And then the only way to get that O is to have 2 of those little i's, 2 of those recessive alleles. So that's the recessive homozygote, okay? So those are all your possibilities of humans, right, are going to be one of those allele combinations, okay? And then the phenotypes are shown there as well. So I love this image because it shows you the genotype on the left of the blood type, your actual blood cell appearance, because I said, remember, those A or those B alleles actually are representing molecules on your cell. So you either have A type molecules or B type molecules. So in both the A and the B case, you have 2 potential genotypes that would give you the same phenotype, right? Which is an A or a B type blood group. So if you're an A type blood or B type, you don't know if you're a homozygote or a heterozygote, okay? And then you have your codominance with the AB, so you have both types of molecules on the cell, right? Or your little I, and you have both recessive alleles, and you have no molecules on the red blood cell, or it's naked, and you are the O-type phenotype group, Okay, so I think this is a great reference for our different genotypes and phenotypes of our blood groups. So now let's do some practice questions with Punnett squares and blood typing. So we have two questions. So the first one is, two parents, one type O, one type AB, have children. What are the children's likely phenotypes? So again, you're going to want to do a Punnett square to get those genotypes, which will give you the phenotypes. And our second question is, you have a mother who is type B, has a type O child. The father accuses her of adultery because he says he's type A and therefore couldn't have a type O child. And so you're answering the question is, could his case stand up in court, or could they have a child with that is type O? So again, go ahead and pause the video and make your Punnett squares, and then we'll go through the answers. Okay, so hopefully you were able to answer these questions, work your way through the Punnett square. So here are our answers on the right hand side. For number 1, if we have a type O parent on the top, they're going to be genotype i. And then if we have a parent that's AB, we have IA and IB on the left hand side. So that means that all their children are either going to be heterozygote type A or heterozygote type B. So again, if we're talking just about phenotypes, 50% of their children will have type A blood, 50% of their children will have type B blood, right? If we're just talking about phenotypes. Now let's go through the second question. This is a little more confusing just by the wording of the question, but essentially the question is, can a mother who is type B and a father who is type A have a type O child? And so our example on the right hand side is yes, if both mother and father are heterozygote A and B, right? So we have father on the top who says he's A, so he could have either two big A's or a big A and a little I, right? So let's say he's the heterozygote, and then same with the mother who's type B. That doesn't tell you whether you're homozygote or heterozygote. So she has at least the IB and probably the little I as well to be able to get an O. So then that gives us a cross, and we see that we do have 1 out of 4 children could be type O, right? If they both get that recessive allele, little I, they will be type O. Okay, so his case would not stand up in court because they could prove that they're both heterozygote blood types and can have a child with type O blood. Okay. So what are some other variations on Mendels laws? So we talked about codominance, we talked about incomplete dominance, right, as being some examples where our dominant allele isn't always dominant. We could have codominance or we could have incomplete dominance, right? So I want you to be able to do some examples of those with Punnett squares, but now I just want you to kind of understand and be able to define some of these other variations. So the first one is polygenic inheritance. So poly meaning multiple. So we have multiple genes that are going to contribute to just one phenotype. Okay, so for example, in humans, skin pigmentation is due to over 3 genes, as well as environmental factors, to produce a very wide range of skin colors. So just for an example on the left-hand side, if we say that it's 3 genes, we have gene A, B, and C, which are all going to contribute to skin pigmentation. So if you have all the recessive alleles, your skin will be very light colored. If you have all the dominant alleles, your skin color will be very dark. And if you are a heterozygote and mix all of those, you'll be a medium shade. Right? So this is where that incomplete dominance comes into play, but now we're talking about multiple genes at the same time that are all going to affect pigmentation. Another example is eye color. Okay, so we do have multiple genes that will contribute or do contribute to eye color, so it's not black and white for eye color either. So again, this idea is called polygenic inheritance. So again, we're not going to do any examples using this because it is very complex with multiple genes taking place and contributing to one single phenotype. But just to have an idea and a definition for what it is. The next one we're going to talk about is pleiotropy, where we have one gene affecting multiple traits. So essentially it's kind of the opposite of polygenic inheritance, because now we have just one gene that could affect multiple things. And again, I'm not going to have you do any examples of pleiotropy, but just to get an idea of the definition of what it is and some real life examples. But again, we're not going to do any Punnett squares or anything like that. But we do have an example of autism and schizophrenia being two completely different traits, right? Whereas they are actually due to the same gene deletion on chromosome 22. And of course, there are other factors that affect autism and schizophrenia, but they have noticed that they're a similar gene deletion. Okay, so one gene can affect multiple traits. So the phenotype being displayed, right? So it's the same gene deletion on a chromosome. So you don't have this one gene. So what happens? You either have autism or schizophrenia, but you say, okay, they're completely different, right? So the phenotype depends on when the disorder is developed. So if you develop your disorder during childhood, you develop autism. If you develop it during adulthood, you develop schizophrenia. So essentially it's the same gene deletion, but it can affect multiple traits. Okay, so you're either going to have childhood autism or adulthood schizophrenia. Okay, so again, very interesting idea, and why, you know, sometimes genetics isn't very straightforward as we wish it were and followed Mendelian laws. So let's do a little bit of review. Okay, so this is what you guys are going to do at home. Okay, so we're not going to go through all of this, but we have already. So you guys are going to be able to solve some problems with these following variations. So we've talked through incomplete dominance and codominance. They're very similar, so I want you to know the difference and be able to do some examples for both of those. We also talked about multiple alleles when it comes to blood typing, right? So multiple alleles in blood typing, I definitely want you to be able to do some problems and Punnett squares with blood typing, right? So those I definitely want you to be able to solve problems for with Punnett squares. So definitely practice those, okay? And then I want you to be able to define the two following terms, but not necessarily solve any problems, right? So know the difference between polygenic inheritance and pleiotropy, okay? So now let's talk a little bit about linked genes. So this is going to be where we take chromosomal basis of inheritance into account. So far we've just been talking about genes that are on either different chromosomes or very far apart on the chromosome so that they are passed on separately, right? We talked about the law of independent assortment. So what happens if we have two genes that are really close together on the same chromosome? Well, they can be inherited together, which is why we call them linked, okay? So this gets rid of the idea of independent assortment. So there's no law of independent assortment. So you don't get 4 separate possibilities, you only get 2 kinds of gametes. So they go together, right? So if we look at our example below, and we have the 2 genes A and B. So they're really close together on the same chromosome. So that means they're going to go together. So you can only have either big A, big B, or little a, little b. So there's really only 2 options or 2 kinds of gametes. Now we have an exception to this rule, and that's in the case of crossover. So remember we talked about crossing over events during meiosis, where they can actually switch genetic material during meiosis, during prophase, when all of those chromosomes are kind of jumbled together and they get very close contact, and they can actually commingle and essentially quote-unquote cross over and swap genetic materials, okay? So this means that normally, if we don't take crossing over into account, we only get those 2 kinds of gametes. And so that means that certain phenotypes may not occur, okay, unless you do have a crossing over event or what we call recombination. So if we have a crossover event, we call it recombination. So let's see what this looks like in comparison to our normal dihybrid pattern, okay? So if we have unlinked genes, right? Say A and B are on completely different chromosomes, we have 4 potential gamete combinations, right? Because we said that they can be swapped if they're on the opposite sides. So that means you can have big A, big B, big A, little b, big B, or little b, little a. So you have 4 different combinations, which then gives us that dihybrid pattern we've been looking at before. So we can cross our 2 heterozygotes together and get that big Punnett square that we kind of went through, but mostly just talked about the phenotype that comes out, our 9:3:1 ratio of phenotypes. So that's what we've been talking about, right? Unlinked genes. So what happens if they're linked, right? So that means you can only get 2 combinations, and that we call a monohybrid pattern, or only 2 potential gametes. So this is where we can, not have certain phenotypes unless we have recombination or crossing over, okay? So this is a great figure I found comparing our linked genes with and without a crossing over event. So we said linked genes are on the same chromosome. That does not mean they are sex-linked. So sex-linked is different, so we'll get into that. But let's look at the top example without a crossing over event. Essentially, we said only 2 gametes can be made because they're going to go together, right? So you can only have big A, big B, little b, right? So what happens though if we do have that crossing over? How many gametes can we then have? So then we get our same 4 options because we have now swapped the big A and the little a, right? And vice versa, however you want to think about it. So you've swapped the two genetic materials. So now you're able to get your recombinant gametes, therefore you have 4 possible outcomes. So if we're talking without crossing over, you only have 2 gametes. With crossing over, you have 4 gametes. Okay, so this is just in the case of linked genes. So now we're going to talk about those sex-linked genes. So we just covered linked genes, which just means that the genes are close by on the same chromosome, so therefore they're going to get passed on together. So what happens with sex-linked genes? So remember, with our sex chromosomes, we have an X chromosome and a Y chromosome. And we said that they are not completely homologous, right? So 2 X chromosomes are homologous, but the X and the Y chromosome are not. So they have enough homology to be able to be paired together, but remember, our Y chromosome does not have as much genetic material. So that means that the genes are then carried on the X chromosome and not on the Y chromosome. So how did they discover these sex-linked genes? Well, there was a man named Thomas Morgan in the early 1900s, and he found that he actually saw a male fruit fly, which the fruit flies are on the right-hand side there. I'm sure many of you have seen them. They're very tiny, but they usually have red eyes. So red eyes is the wild type, and he found a male with white eyes, so kind of strange, and he was trying to figure out, how did I get a fruit fly, a male fruit fly nonetheless, with white eyes? So what was his prediction? What did he experiment? He took one of those white-eyed females, which he called little r, right? So we have 2 recessive alleles for that gene, and he crossed it with a female, which he called big R for the dominant red allele. So this is what he was predicting, right? From what we know of Mendelian genetics, this is what he predicted, right? So his prediction was, if he crossed that parent cross for the F1 generation, he'd get 100% red, right? They'd all be heterozygote, big R, little r, and they'd all be red-eyed. And then he said, well, how about my prediction for the F2 generation? If I cross 2 of those heterozygotes together, I will get a 3-to-1 ratio red to white, right? That should be our classic Mendelian genetics. Well, that's not what he got. Surprise! In his F2 generation, all the white-eyed individuals were male. So if it was indiscriminate, meaning not sex-linked, he would have some white-eyed females as well, right? But he found that the only individuals that had white eyes were male. Okay, so how did he get this? What happened? So he posed the question, why were all white-eyed individuals male? Well, the answer is sex-linked genes, but how do we express that when we're talking about Punnett squares and alleles? So we said that in sex-linked genes, the genes are on the X chromosome only and not on the Y chromosome. So what he did was he said, okay, so the parent generation, the red-eyed female had to have been an X, so we say the X for the X chromosome, XR for dominant R allele, right? And then he crossed that with a white-eyed male. So he had to have the X chromosome had to be recessive, so it had a little r, right? And then we have the Y chromosome. So the Y chromosome again does not have the gene on the chromosome. So it's only the X chromosome. So what did he get then? Okay, so here's our Punnett square. We've got XR dominant on the left-hand side for the female, and across the top we have XR little r, right? And the Y. So then we notice that all the individuals are red-eyed, even the males, right? So we have two females that are heterozygote because they have big R, little r, and then both males are red-eyed, right? So, okay, so that still kind of fit that 100% red-eyed, right? But what happened with the F1 generation? So now he's crossing that heterozygote red-eyed female, XR big R, XR little r, right? With a red-eyed male. So all the males from the first generation were red. And this is what he got, right? So now we have the females are all red-eyed, right? One homozygote, one heterozygote. But here's where our white-eyed female or white-eyed male comes, excuse me. So 50%, right, are going to be white-eyed of the males, right? So you still get some red-eyed males, but there's where that white-eyed phenotype comes out, right? Okay. So if we're talking about sex-linked genes, why are more males affected, right? For our example of the fruit fly eye color, he only saw males as having the white eyes. There were no females with white eyes. So just in general, we're saying that, the X chromosome has a lot of traits that are unrelated to the sex of the individual. So females are going to carry 2 alleles for any given sex-linked trait, right? So they have to have 2 alleles because they have 2 X chromosomes. Whereas males only carry 1 allele for that sex-linked trait. Therefore, it's much easier for them to express that recessive phenotype because they only need the 1 recessive allele. Okay, whereas females, they have to have both recessive alleles. So when we're talking about sex-linked disorders, they really affect mostly males, and that's why, right? So females need 2 recessive alleles to have the disorder because all the homozygote dominants are all going to be the phenotype, dominant phenotype, as well as the heterozygotes, right? So that's why we see males affected more by these sex-linked disorders. So let's talk about one example of a sex-linked disorder. So obviously there's many sex-linked traits that don't necessarily mean a disorder, right? But there are some disorders such as color blindness. Now, if you know anyone that's colorblind, or if you're colorblind yourself, you're probably male, okay? So let's look at an F2 generation, meaning that we're crossing a heterozygote female with a dominant a male, okay? So that means that both the female and the male are not colorblind. So we say C for colorblind, right? So big C is going to be not colorblind. Little c is going to be colorblind. So female on the left, she's heterozygote because she has a big C, little c, but she's not colorblind. And then the male is big C because he's not colorblind and a Y, right? So what crosses do we have? Well, our 2 females are both not colorblind, right? Even though one's a heterozygote. But then we do have one of our 2 males that is colorblind, right? So if we look at what the percentages are, what percent of females will be colorblind? If we look at that. 0, right? We said 0. They're both not colorblind. Now, what percent of males will be colorblind? We said 1 of 2, right? So 50% will be colorblind. Now, what percent of all the children will be colorblind? And that's 1 in 4, right? Or 25%. So here are the answers, right? 0, 50, and 25%. So there are some sex-linked disorders that are a little more serious than, color blindness. We can function just fine with color blindness, but hemophilia is an abnormal allele for blood clotting factor. So if you are a hemophiliac, you bleed excessively when injured, which can be quite serious, especially way back in the day when medicine maybe was not up to snuff. So there is a very interesting history to hemophilia in the European royal bloodlines. And if we look at some of these pedigrees, so a pedigree is a family tree that actually uses phenotypes to predict phenotypes and genotypes of family members. So how a pedigree works is usually you are a square if you're male and a circle if you're female. And then depending on the color of your box or circle, that shows you the phenotype and genotype. So if you're a solid color, you are a homozygote for that trait. And usually if you're the dark color or bright color, you are homozygote recessive. Okay, so you have the recessive trait such as hemophilia. Okay. And then if you are half and half, you're a heterozygote for that trait. So if we're talking about sex-linked disorders, females, right, are going to be heterozygote carriers, essentially, of that recessive disorder. So if we're talking about hemophilia and we see these three circles here, starting with Queen Victoria, who was a queen of England back in the day, who had many children, she was a carrier for hemophilia. So she did not have hemophilia, but she had multiple children that became carriers for hemophilia, which then married into a lot of royal families in Europe, and then they proceeded to have children, and any male children that they had a high percentage chance of getting hemophilia. And so this is what happened in this specific family tree, which was in Russia. So one of her granddaughters married the Tsar of Russia, and she was a carrier for hemophilia, and they had a son who became a hemophiliac because he only needed that one allele, one recessive allele from his mother. So very interesting kind of disorder that worked its way through a lot of royal families in Europe because of Queen Victoria, who essentially passed on this recessive disorder, this sex-linked recessive disorder throughout Europe, and they actually blame the fall of Russia somewhat on this disorder. So just if you want to know more about hemophilia and the royal families and how they kind of blame it all on the fall of Russia on hemophilia, there is a really great article that I've posted on Canvas as well as in the PowerPoint slide here, and it does have some examples at the end of the article as well, some problems to kind of work through that are good. So check it out if you're interested and want to know more about the royal disease, hemophilia. So now let's do a little bit of review on kind of these sex-linked disorders. So we're going to talk about colorblindness. So for the first question, we have two people with normal eye vision. So two people that have normal vision have a son with color blindness. What is the likely genotypes of the parents, right? So how do we get that colorblind son? Okay, so that's question one. And then question two, if you have two parents, have a colorblind daughter. So again, now we have a daughter that's colorblind. What has to be their likely phenotype? So again, phenotype. So we're going back up to phenotype. But you can either kind of work your way through this, or you can make some Punnett squares, right, with these X-linked genes, okay? So go ahead and pause the video and then we'll go through the answers. So let's go through number 1. So what is the likely genotypes of the parents to have a colorblind son? So we know that they are both normal-eyed parents, right? So mother has to be heterozygote for colorblindness, meaning she has to have a big C and a little c, right? Because the father has to have a big C on his X because he is a normal normal vision person. So those are the answers. So mother has to be heterozygote. Father is dominant, right, for colorblindness, for not colorblindness. Okay. And then the second question, so the mother is not colorblind, right? But she is probably a heterozygote. Sure, she has to be a heterozygote. I mean, I guess she could be colorblind as well, but just because that's more normal, she's probably a heterozygote for colorblindness, right? So she has the one recessive allele. But the father has to be colorblind to be able to get a colorblind daughter, right? So he has to have that colorblind allele on his X chromosome to pass that on to the daughter. Okay. So that's it for the patterns of inheritance 2. Hopefully these weren't too confusing. If you need some more practice questions, let me know. I may post some on Canvas for you guys to work through. And I will also post a good video on kind of sex-linked genes and linked genes, period. So I will see you guys. If you have any questions, come to office hours, and I'll see you guys next time.